for a graduate program, and there he studied sleep and the role of astrocytes. So astrocytes are known to release some neurotransmitters, and he knocked out that process, and it turned out that that really has a profound effect on sleep. And there, so after that, he went to residency program at MGH, working together with Emily Brown and Matt Wilson. He continued to study sleep, but also getting more interested in attention and executive functions. And he did one of the very first recording of ensemble neurons in the thalamic reticular nucleus, which is a very mysterious structure surrounding the thalamus, and showed that the activities are modulated depending on the sleep and cognitive demand, and they regulate these processes. And after that, he went to NYU as an assistant professor studying his own lab, and he continued to study and extended his study in the area of attention and cognitive processes, particularly focusing on the thalamus and in mouse models. And he continues to publish a very influential, very interesting work. So I'm very excited to hear your his studies, recent studies. So thanks for coming.
Thanks now for this very generous introduction, and thanks for the invitation to talk to you today about what we do in my lab. And my talk will really focus on trying to tell you about the sort of the theoretical framework that's budding that tells us something about how the cortex and thalamus interact in the context of cognitive control and flexibility. So we're interested in this idea of cognitive control, the ability of the brain to control our thoughts and action plans based on an internal model of the world, and the flexible ability to switch between these constructs based on context. The particular cognitive process that we focused on to understand these processes is goal-directed attention. Oh, this is not working? Oh. Sorry. Okay. Okay. It's better now? Okay. All right. So the particular process that we've been interested in is this idea of goal-directed attention, or top-down attention, which is the process that we imagine allows us to pick out a single musical instrument from an orchestra, or to eavesdrop on different conversations in a crowded room without moving a muscle. So the way we imagine this process to work, and this is based on a number of studies, mostly in humans and non-human primates, is that the goal-directed signals are generated in frontal cortical areas, mostly the prefrontal cortex, and by projecting back onto sensory cortical areas, the PFC can amplify and suppress sensory features according to the behavioral demands. Now, this process has been studied extensively in monkeys. People like Bob Desimone, Earl Miller have worked out a lot of details surrounding this process. When we started studying this, what we ended up, I think, sort of contributing to is the understanding of subcortical processing in this process. We focused on the thalamus, and we've isolated two different thalamic functions that contribute to goal-directed attention. So early work in the lab focused on the sensory part of the thalamus, areas like the geniculate, the LGN, and the MGB. And what we found is that the sensory thalamus can be a locus for attentional and control, meaning that as soon as information gets from the retina, from the somatosensory system, from the auditory system, it can be gated according to behavioral demands at the level of sensory thalamus. And from that perspective, the thalamus is still a relay, but it's more of a gated relay. It can be gated as a function of what one cares about. Now, what I will spend most of my time today talking about is a distinct thalamic process that has to do with the non-sensory thalamus, areas of the thalamus that don't receive their primary driving input from the periphery, but rather from the cortex itself. And the prime example I'm going to talk about today is the mediodorsal thalamus, which gets most of its driving input from the PFC itself and projects back onto the PFC, not so much to relay any type of peripheral input, but more so to configure cortical representations or these attentional signal according to the context at hand. And I will tell you some details about it throughout the talk. So this is a distinct idea from relaying information and has to do more with the gating of internal representations that are ongoing in the cortex according to the context. So let me make this idea a little bit more crisp and put it in a computational framework of how we understand the classical roles of the thalamus. So what is the way we understand thalamic processing generally is based on how the LGN works. And the LGN receives its primary driving input from the retina and projects to primary visual cortex. And there are particular types of features that it has both on its input and its output that makes us think that it operates like a relay. The first is that the input from the retina is highly non-convergent and therefore the weights don't really change after the critical period. So the types of computations that happen from the retina to the LGN are limited. And therefore, the representation, the receptive fields in both areas are center surround and roughly similar. Now, the output from the LGN to V1 is primarily excitatory. And the way we imagine that to work is that V1 neurons build their receptive fields based on conjunctions of LGN responses. So if you choose the appropriate types of LGN inputs, you can end up building these direction selective and orientation selective responses at the level of V1. So not much happens at the level of the input, therefore a very limited computation at the level of retina to LGN, and mostly a relay function from the LGN to V1. Now, for a structure like the MD, both the inputs and outputs are different, and that's what I will try to convey to you today. The inputs are the inputs are from the PFC, the outputs are to the PFC. There's quite a bit of evidence for convergence from the PFC, and those weights can be adjustable. Therefore, bona fide computations can happen at the level of MD, and one can observe conjunctive representations in the thalamus that are based on sort of distributed representations in the cortex. And the output from the MD can also have different features than that of the LGN. A subset of these outputs can be primarily inhibitory. And why is that? Because the way responses or the way representations are built in an associative cortical area like the PFC are actually pretty different than that of V1. So representations in the PFC, like attentional or cognitive signals, can be built based on inputs from the PFC itself or other cortical areas, but are quite flexible and task dependent. So you can have a choice of how these receptive fields can be built according to the context. What we imagine happens is that the contextual representation gets computed in the thalamus, and the thalamus uses that contextual representation to regulate which input PFC representations or cognitive signals are listening to. So ultimately what the thalamus ends up doing is regulating the functional connectivity within the cortex to configure the representations or the cognitive output signals according to the task demand. And that's what I will talk to you about today. So my talk is going to be divided into 3 different parts. One, I'll tell you about how we managed to train animals such that they build hierarchical representations that switch according to context. And then I'll tell you about the role of the thalamus in regulating and arbitrating between these different contextual representations. And then I will tell you something about the generality of these functions beyond mice. Okay, so the task that we developed in mice in order to ask questions about building abstract hierarchical representations, PFC representations, is the following task. It's a 2 alternative force choice task in which an animal chooses on each trial between conflicting visual and auditory stimuli. These are very, very simple stimuli with low feature complexity. So it's a visual flash that can appear to the right or the left of the animal, or an auditory sweep that tells the animal to go to the right or the left. On each trial, the animal gets a different spatial configuration of these stimuli and has to choose whether to go to the right or the left, depending on whether it's a visual trial. Same thing, right to the left, whether it's an auditory trial. How does the mouse know which one of these two stimuli is the valid stimulus? We cue it on each trial with this arbitrary cue. It's a simple 100 millisecond noise burst that the animal learns over a period of several weeks to know that one of them is associated with a visual stimulus, whether it comes to the right or the left, or an auditory stimulus, whether it comes to the right or the left. So the animal gets one of these cues randomly on each trial, has to presumably hold the cue in mind over a delay, and uses it to map onto the appropriate target stimulus. So this task basically captures the essence of what we imagine goal-directed attention to be. It has a cueing component that engages the prefrontal cortex and the executive component of the mouse's brain, and then a sensory component that has to do with arbitrating between a broadband visual or auditory stimulus. So the fact that we got this task to work in mice, and it works fairly well, and we've built regression models to determine how much does the animal use the cue. So we have some idea of the engagement of the strategy that the animal actually uses to solve this task. So we're pretty convinced that the animal does use a strategy of taking the cue, holding it in mind, mapping it onto the appropriate task. And that basically allows us to figure out what the causal engagement of different brain areas in solving this task. And the first thing that we determined is that just like primates, including humans, the PFC, the mouse uses the PFC in this component of the task, what we call the executive part of the task, so the cueing and the delay. If we silence the PFC on a trial by trial during these phases of the task, the animal basically guesses. It's equivalent to catch trials in which we give the animal no cue. But if we silence the PFC during the presentation of the target stimuli, the animal can do the task just fine. Okay, yeah?
If you remove the PFC altogether,
Yeah, it's a good question. We haven't done that. We haven't done that.
what your prediction would be they would be guessing then, right?
They would, so it's a hard problem to, so my guess would be that they wouldn't be able to learn the task. They wouldn't get to a trial by trial. So the way we shape these animals is that we train them on the two tasks separately, and then we have them do it in blocks, and then we shift to trial by trial. My guess is that they would break somewhere at the level of block to trial by trial transition.
But if you after learning, if you just remove it altogether, not transiently silence, you would think they would just keep guessing then.
My guess is that they wouldn't be able to do it trial by trial. They may default to picking one of these stimuli. Yeah. Because these stimuli are of low feature complexity, what our experiments have shown is that the way the task gets solved neurally is as follows. The PFC simply arbitrates between the visual and auditory pathway at the level of sensory thalamus. So in attend to audition trials, the PFC sends this control signals that ultimately changes the gain of visual flow at the level of LGN. And in the attend to vision trial, the PFC changes the gain at the level of the MGB. That's basically the model that we have. And consistent with that model, I'll just tell you briefly what the data are. If we record from the LGN or the structure that now mentioned, the thalamic reticular nucleus that's specifically associated with the LGN, we see changes in spike rate that are dependent on how the animal was cued, even before the stimulus is shown, so anticipatory changes in spike rate that are completely eliminated when the PFC is silenced. So that's the evidence that this is the strategy and this is the neural implementation. But then we simply ask the question, what exactly are the neural codes underlying the encoding and the maintenance of the cue information over time, given that we think that's what the PFC does? So we recorded, so just to remind you, this is the part of the task that we think the PFC is engaged in, the cue and the delay. And this is an example of a PFC neuron that shows task selective responses. So this is just one neuron, It's a regular spiking neuron, so a putative excitatory neuron, that and 12 to 15% of neurons, regular spiking neurons in the PFC, do this. So the trials are separated into the two cueing conditions. 0 is, so these are the rasters. Rasters are basically each line is a trial and each tick is an action potential. 0 is the time that one of the cues are presented. And in this particular experiment, 500 milliseconds is when the targets are presented. So what this neuron shows is a temporally sparse increase in spiking at this brief moment during the delay. And it shows it for one of the cueing conditions, not the other, right? If we look at multiple neurons in the PFC, we see that they tile the delay period. So each neuron has this type of sparse temporal response at a moment during the delay. And in this task, there are two different functional subgroups in the PFC that emerge. I mean, obviously, this happens during learning, because even when in well-trained animals, when the animal doesn't follow the strategy of cue, if the animal doesn't follow a strategy in which it's utilizing the cues, we don't see these responses. So these are dependent not only on training the animal, and the animal following the right strategy. Okay? So these basically look like the sparse responses that people have observed in the songbird HVC, for example, where the model for that is a synaptic chain, or at least a feed-forward mode, right? I'm not going to say a synaptic chain, but at least activity is handed off from one mode of the network to the next, to the next, to the next, to the next, right? We can actually test whether this is a viable model by looking at spike time cross correlations between neurons in the task and ask, if neurons are categorically co-tuned, meaning that they care about the same cue, are they more likely to show these putative synaptic synaptic connections? And the answer is yes. So when we look at these, short latency spike time cross correlations between pairs, they're much more likely to exist between neurons that show these task selective responses, and much more likely to exist between pairs that are categorically co-tuned and that have overlapping temporal fields. Okay? This is exactly the same answer from a generalized linear model in which we predict the spiking responses of neurons based on the responses, based on spikes from neurons recorded at the same time. And this is basically exactly the same answer. Neurons that are likely to encode the same cues are more likely to be connected to one another than neurons that encode the different cues. So the model that, yeah.
Do you see this on air trials too, or are they just?
It's a great question. So it depends on why the animal is making an error, right? So, and it depends on how many error trials we have. So in some sessions, when we have sufficient number of error trials to be able to look at these types of phenomenology, we see that these responses show up when the animal makes basically, we see the tuning in the opposite cueing condition on error trials. So if the animal thought it was attend to vision, but it was attend to audition, we see this type of tuning. And so we think that the majority of errors on those sessions can be attributed to misattribution of the cue itself. So the animal thought it was high pass when it was low pass, for example. But the model that we have based on this data is a simple feed-forward mode in the network that maintains the memory over time. So it's basically when the animal gets, yeah.
I think you said that when you show the high pass or low pass, when you play in the absence of the context, you don't get these responses.
I haven't said that, but that's a great question. I will get to it at, yes, but we don't get that outside of the task. Yeah. So basically, this is an input-driven regime in which the activity propagates are maintained in the network in a feed-forward way. But the one thing that I want to point out here is that based on the spike rates of these neurons, these neurons with temporal fields, we can decode what the animal's thinking, what cue was encoded. I mean, we can just apply simple linear decoding on the data and figure out what the cue was. Importantly, there is no hint of a preparation for movement at that point in time, because the movement itself is conditioned upon the particular spatial configuration of the targets, which at this time the animal has no access to. So this is not a preparation for movement task. We think this is some abstract variable that the animal is maintaining in its mind over the delay. That tells us something about how much mice can abstract from their environment. But this became the next question. How abstract are these representations? Now we have high pass noise, low pass noise, which to us mean attend to vision, attend to audition, right? But from the mouse's perspective, it's unclear how abstract these representations are. Are they memories of the cues, or are they actually task rules, right? We don't know based on what I've shown you. So what we ended up doing is training the animals to do this task based on 2 different colored lights, have them run through a session in which they're playing the auditory game, then switching to the visual game. We observe the same neurons in the same session and ask, are the neurons, do they care about the cues, or do they care about the meaning? Oh, sorry. Do they care about the meaning of those cues as task rules? And it turns out we have both kinds of responses in the mouse PFC. A subset of neurons care about the cues. They care about 1 out of the 4. And we have them for all 4 categories. And a different subset of neurons care about the conjunction of these cues as they pertain to meaning. So there exists a subset of neurons in the mouse PFC that don't care how exactly you cue the mouse. They care about what the cue actually means. And because we end up recording these neurons at the same time, we can fit them into multi-neuron generalized linear models, figure out functional connections. And what our data suggests is that neurons that are cue selective and encode the same cues have strong lateral interactions with one another. And both cue sets that mean the same thing have feed-forward connections to these cue invariant or meaning neurons. So we think this type of hierarchy is built in the PFC, where the cue ends up, arbitrary cues can be transformed into meaning through this type of network architecture. Now, we have some idea of what these cue invariant neurons are in this task, because we imagine for each type of task, the transformation will end up recruiting some output pathway from the PFC that would tell the rest of the brain which sensory motor transformation to regulate. In this particular case, it's attend to vision or attend to audition, or ignore the distracting modality. So what we end up studying is the pathway by which the PFC can influence activity at the level of sensory thalamus, given that there are no direct projections from the PFC to the sensory thalamus. And based on 2 different types of studies, one is anatomical traces out of the PFC and from the sensory thalamus, as well as functional dissection of the different PFC output pathways that would end up making it to the thalamus. What we concluded is a pathway in which the PFC can influence activity at the sensory thalamus through a basal ganglia pathway. And this has to do with the tail of the striatum, the posterior part of the striatum, that is also segregated according to modalities. And I'll just show you one piece of data that supports this idea. If we tag neurons from the tail of the striatum that project to either the visual or the auditory striatum, we see responses in this PFC neurons that confirm the allegiance of these neurons to a particular type of cue. And those end up being the meaning neurons in the PFC. At least a subset of meaning neurons in the PFC are ones that project to these sensory striatal territories and end up regulating thalamic transmission at the level of sensory thalamus. So just the first part of this talk, the mouse PFC can build hierarchical representations in a task-relevant manner. I hope that was clear. Sensory selection for broad stimulus features can be performed at the level of the thalamus. And the striatum is involved in transmitting PFC output to sensory gain control, relevant for attention. So that's the first part of the talk. And Now I'll turn into this medial dorsal thalamic involvement. And the way we got into this question is just like Florian asked. We just wondered, are these feed-forward representations in the PFC, are they contextual? Does the animal have to be in the task in order for these feed-forward modes to exist? And the answer was yes. So we have the animal engaged in the task. We present the cues. We see these temporal fields in the task. We take the animal, give it the cues outside of the task. We get nothing. Okay. So then we said, okay, well, how is this done, right? The simplest sort of model for this is that the cue is somehow gated. It doesn't get to the PFC in the first place. And where does the cue come from? Well, the sort of standard thalamocortical model is it comes from the thalamus. And the biggest thalamus that projects to the PFC is the medial dorsal thalamus, right? So that was a simple model. And then maybe there's just a gate here. It doesn't make it into the cortex. And that model completely failed based on the fact that when we suppressed the MD thalamus during the cue period alone, there was minimal impact on behavior and physiology. Okay? So that told us that the cue information cannot possibly come from the MD thalamus. But then there was something pretty surprising because we can suppress the thalamus at different phases of the task. When we suppressed it during the delay period, the activity in the cortex fell apart. So what happened is, so here I'm showing, a measure of the peakiness of neurons and comparing the laser off trials to laser on trials when the thalamus is suppressed. And what we see basically is that the early responses in the cortex, we still get them, these cues, these responses following the cues, but the later responses in the cortex are diminished when the thalamus is suppressed. So that was pretty surprising. So the thalamus is not a source of information, cue information to the cortex. It's doing something else. What is it doing? So we recorded from the thalamus itself. And what we found is that the thalamus shows these transient responses, but individual thalamic neurons don't care about what the cue was. So they have these task selective responses, but they're not cue selective at all. So that sort of confirmed the idea that the thalamus is unlikely to be a source of inputs to the cortex in this task at least. And this type of thalamic phenomenology is completely dependent on, so there's no cue information. It's completely eliminated by suppressing the PFC, even in the cue period. So what we think happens is that cue information comes to the cortex and then gets funneled into the thalamus. And to account for these types of responses, we think that there's convergence from these two different chains into the cortex onto individual thalamic cells, accounting for their responses in the task. So that's the model that we have for what this thalamic activity, how that's generated. But why is it that you need that? Okay. So what we found so we asked the question, okay, the input to the thalamus seems to be from cortex, right? What is the output actually doing if it's not cue selective? So the first evidence is that it's primarily suppressive. And this is based on simultaneous recording in the cortex and the thalamus at the same time, and trying to explain spike variability in the PFC based on inputs from the MD. And what we found is that the coupling was mostly inhibitory from the thalamus to the cortex. And when comparing that to a sensory thalamocortical recording, this was the total opposite. If we record from the LGN and V1 or MGB and A1, the coupling is mostly excitatory. Okay, so that was pretty surprising. The thalamus is suppressing the cortex. All right, but this is correlational. So just to sort of nail this point, we did this direct causal experiment where we took a classical sensory thalamocortical loop and this MDPFC loop, and basically asked if we just turn on the neurons in both structure, what is the impact on their cortical targets, right? And we used these stabilized step function opsins, which are basically just a current injection into individual neurons. And we validated that those tools work just the same in the MGB and the MD. So they give us a sort of a step in spike rates. But the impact on these manipulations across the two different cortical targets is completely different. While A1 responds to this step in spike rates in an equivalent step in spike rates, so excitation here, E here ends up being E here. E here ends up mostly suppressive here. Okay? So that was pretty surprising, that you can activate a thalamic region and get mostly inhibition in the cortex. But that's not the only thing that you get. You get a different effect, which has to do on how well the neurons talk to each other in the cortex after a thalamic activation. So this we can measure through coupling filters in the cortex itself. We can look at A1 pairs and PFC pairs that have roughly the same type of coupling strength. Activating the thalamus, the MGB, suppresses lateral connectivity in A1. Activating the MD enhances lateral connectivity in the PFC. And that's what we think is the signal that allows for the chain propagation over time. So the model that we have for this is that we have in the PFC of the mouse, in this task, the memory is maintained through these feed-forward modes of the network. It's input-driven. The thalamus receives these convergent input from the cortex, and it sends out two different outputs. One that enhances the lateral connectivity between the neurons, and we have some ideas about what the biophysical mechanisms for that is, and I'm happy to talk to you about it afterward. And another that mostly drives inhibition, such that when you have balanced amplification of these memories, and that when you have a chain propagating for one memory, the other memory doesn't get activated. So you have both amplification of the memories themselves, but balance in that you're driving inhibition so that the memories don't talk to each other. Why was this system built that way? I'm not entirely sure, but I think I can tell you something about how this same architecture gets utilized for more complicated tasks, if we have time for me. Yeah, sorry, did you have a question?
So one of the ideas is that the memory of the cue is somehow kept alive in the prefrontal cortex.
Yeah.
And it's going through this loop through the MD.
Not in terms of content.
I see.
Not in terms of content.
So the MD would actually be during the delay, it would be silent, right?
No, during the delay, it would be on. It would show these non-specific chains.
But it's only it's either inhibitory directly or it's targeting an inhibitory neuron, the purple one.
Oh no! So these are just. So we have a model for this, and these end up being just gain on the synaptic connections within the cortex.
Yeah.
Okay. Sorry, this was not clear.
Can you just clarify?
Yeah.
MD is non-specific targeting the blue and red, right?
Yeah.
How can you get one activated and one?
It's input driven. It's input driven.
So it's kind of one-take-all.
It's a winner take all, and it's basically enhanced by this inhibitory mechanism. Now, this is non-specific in this particular task. So let me tell you about two different extension of this model that would show some selectivity, and that selectivity may be happening through learning rules at the level of thalamus and cortex. You can get some selectivity. This is the vanilla generic sort of model, but you can imagine that you can target specific types of interneurons in the cortex if there was learning on these connections. And this was learning on these connections. And there's evidence that there is learning on this connection from two different experiments that we've done. One, we went back and studied this process in the flexible switching between the two different games. And then the other one has to do with giving the animal ambiguous cues that it has to integrate over time and showing that the thalamic signal becomes even more nuanced. And I'll tell you about that. Okay, so the first experiment in which this becomes extremely useful is in this high pass, low pass versus UV green task. And again, in this task, we don't see cue-specific thalamic responses. What we see is game-specific responses. So basically, if you remember, the conjunctions in the cortex that were being formed were about the meaning. Here, the animal is forming a conjunction that has to do about the game it's playing. If it's being cued as high pass, low pass, that's what shows up in the thalamus. When the animal plays UV green, those cells become silent and different cells take over. So basically the thalamus tells you which block you're in. Now, is this a modality bias? Auditory versus visual game? And the answer is we can test that. We can have the animal play arbitrary games, right? We can have the animal play a green high pass or a low pass UV, right? And ask, does the thalamus follow this thing or does it follow this thing? And it follows this thing, right? So and the reason why I think that there's learning happening on these connections and that learning is pretty fast is because the previous day the animal played this game, right? So if this was hardwired or if this was long-term memories, why are these cells not showing up on this day, right? So the fact that these things get configured when the animal plays the game suggests that they are being built on the spot.
Could it be that the basic ganglia influence this?
Oh, absolutely.
So the plasticity of the switch is actually in the basic ganglia?
It's possible.
It's different.
It's possible. But there's got to be some plasticity somewhere that allows for, yeah.
Learned habit, a learned task that is represented in the basic ganglia.
It's possible. I mean, yeah, it's possible. So there's one type of representation that we see, which are these peaky thalamic cells, but there's also that we call transient cells, and there are these more persistent activity that we see across the entire delay in the thalamus in this task. And again, we can look at input-output proxies for these neurons based on multi-areal generalized linear models. And what we basically find is that these neurons are the ones that are more likely to drive the lateral connectivity in the PFC, and these are the neurons that are more likely to drive FS neurons. And in fact, if we actually record from FS neurons in the cortex, we see that their activity looks very similar to these PFC, these thalamic neurons that we record from. And it turns out that these two neural populations are differentially sensitive to optogenetic tools. So we can titrate the optogenetic manipulation in a way to get one but not the other. And therefore, we can use this to selectively target these two populations. And yes, it turns out that when we selectively target this population, we can selectively target these contextual representations that we find in FS cells. So we think that FS cells that are contextual in the PFC are mostly driven by these persistent MD populations. So that basically allows us to slightly refine the model in which we have a single thalamic cell driving both inhibition and amplifications into two different types of cells, each one specialized for one of these two different processes. And again, why do we need that? Well, in this task in particular, where the animal is switching between different cue sets, and both of them mean similar things, what the thalamus actually is doing is quickly encoding the context, right? Whether it's because of plasticity at the level of basal ganglia or on these corticothalamic synapses, unclear. But by quickly computing that context within a few trials, what it does, it amplifies the context-relevant representations of the PFC, suppresses the context-irrelevant one, and allow for these input-output transformations to happen efficiently and basically lowers the noise on this decoder, if you want to think about it that way. So, and then when the animal shifts, again, this takes a few trials. This computes, uses it to suppress this one, so the learning on this connection becomes faster. If this was true, then suppressing the thalamus, even in the cue-specific period where normally in the high-pass, low-pass task doesn't do anything, should delay this process, and that's exactly what we see. So when we have the animals switch from one game to the next, it takes them about 10 trials to get back to asymptotic performance. This would be the visual game. We switch to the auditory game. It takes them about 10 trials to do that. And that may be because they're mice. I don't know. It doesn't take people 10 trials to switch on well-learned cues. But if we suppress the thalamus during the cue-specific period in a well-performed game, not much happens, but the switching takes longer. And this is, in the neural recording, this is consistent with a delay on the emergence of these cue invariant or their meaning representations in the PFC. They can still do it, but it takes longer. So it depends, right? So here, the manipulations is interleaved. So there are enough trials in there for them to be able to build the representation. Now, if we suppress them on every single trial, they never switch. So that's one example of where this architecture, this balanced amplification through the thalamus, ends up being useful. And then this is the potentially the more general case, because this task is as hard for mice as it is for humans, I think. And this is where, rather than presenting the animal with either high pass or low pass, we present it with a sequence in which the animal has to integrate evidence in favor of low pass or high pass, right? And we can titrate the difficulty of the task by making it closer to threshold. And then we can ask the question of, what is the thalamus? Is this noise control function of the thalamus engaged in something like this? And the answer is yes. So I'll show you the model and then I'll show you the data. The idea here is that if you get both with slight bias towards one, there's a process of refinement that takes place during the queuing period, and only one of the chains ends up winning. So this is the evidence. First of all, mice can do this task, which is a miracle, right? And on its own, that mice can integrate evidence towards a rule was extremely surprising. And suppressing the PFC during the queuing period, regardless of the difficulty, regardless of the net evidence on that trial, eliminates the performance, just like we saw in the basic task. What does the PFC actually do in this task? Well, if we look at neurons, what we see is that the PFC neurons, they still show these feed-forward modes. They're broader, but their selectivity increases as a function of the net evidence. So we have more selective responses later on during the queuing period. And this emergence of selectivity changes as a function of the net evidence. So in easy trials, it happens faster than in hard trial, and we can quantify that. So they basically get to a clean representations faster in the easy trials than the hard trials. What does the thalamus do? Well, when we suppress the thalamus, we actually see something quite different. Its effect on easy trials is very limited, but its effect on hard trials is much, much stronger. The thalamus, again, shows no cue-specific responses. Its responses are conjunctive, just like we saw before. But now, they're not on and off. They're graded responses as a function of the net evidence. The harder the trial is, and presumably the more mixed the cues are, the stronger the responses are in the thalamus. And suppressing these responses during the queuing period makes selective responses in the PFC less selective. So basically, that's the model we have. We suppress this thing, and now sort of weak evidence is able to trigger a working memory chain, and the animal is confused, doesn't know what to do anymore. Okay, so the conclusions from this: under certain conditions, the MD thalamus does not operate as a relay. I hope it was clear why I'm saying this, that there's no confusion about why I'm saying that this is not a relay. And that thalamic inputs regulate cortical interactions in a task-relevant manner. The mechanisms are still to be discovered, but include cortical inhibition. Do I have time for just one more thing? So I'll just tell you about the generality of this thing, okay? And the generality is by studying a similar task, basically the same task, in this guy, the northern tree shrew, which is basically you can think of it as a cross between a mouse and a monkey. So it sounds like a South Park episode, but no. So these animals are periprimates. So they, until 10 years ago or so, they were categorized as primates, but they were demoted. I don't know why. But basically, this animal is the size of a rat, but it has a brain much larger than that of a rat, and there's a huge expansion in its frontal cortex. So and it has on Nisselstein at least, it has this layer 4 prefrontal cortex. I don't know what layer, humans have that too. I don't know what it does, but for whatever it's worth, we just picked that because because of two things. One is it was available. So this is a collaboration with David Fitzpatrick at the Max Planck, and David has used these animals to study V1 because it has a similar primate organization like V1. But people haven't explored the prefrontal cortex in that animal, and people haven't really tested how cognitive this animal is. So what I can tell you is that this animal is very cognitive. I mean, compared to the rodents, we can put it in a box, and in a day, it'll figure out what the task mechanics are. You know, in a couple of trials, it'll figure out, oh, you need me to poke, rather than mice or rats that take, you know, a few days to figure out that what we want them to do is poke. These animals figure it right away. And their performance, they learn the task faster, their performance is a lot more robust. And it turns out that the...
Can I just interject? So your definition of cognitive, or your metric of cognition,
Yeah.
is figuring out what humans want it to do.
Yeah. From a human cognition...
Yes, no, thank you.
No, well, I don't...
Talk about that later.
Yeah.
Yeah, I mean, yeah. So from a human cognition's perspective, these animals do well. You know, from a, I don't know, a general universal theory of cognition, I don't know. So, but here's the thing. So there, in the integration of evidence task, according to the high pass or low pass, they start showing much more, persistent representations in the PFC. Ramps, like people have observed in primates. And the ramps are, the slope of the ramp is a function of the degree of evidence presented. Their working memory representations in the delay period are much more sustained than we see in the mouse. So their activity patterns in the PFC are starting to look a little bit more like what people have seen in monkeys for pretty much the exact same task. But what we see is that in the thalamus, the representations are still conjunctive. And the thalamus still cares about how difficult the task is. So despite the differences that might have happened during our evolution, maybe more recurrence, more feedback, the things look a little bit different in the cortex. The spirit is exactly the same. I mean, these are ramping towards telling what the working memory is. And the working memory, yes, it looks a little bit different, but I imagine that the algorithm is the same. I mean, the animal is outputting a signal to control sensory processing downstream. But the thalamus is doing something very, very similar. So, So when one worries about how much mouse neuroscience is being done, I mean, I think that's reasonable to worry about. But I think I think we have a good chance at figuring some interesting things by studying the mice. So, so the conclusion: thalamic reconfiguration, prefrontal representation are general across tasks. And despite differences in cortical substrates, frontothalamic interactions described may generalize beyond rodents. Just to thank the people who contributed to this work, and the collaborators, and the support. And thank you for...
Amazing.
Thank you.
Did you, for your cueing, you use either auditory high pass, low pass, or the UV versus blue? You thought about using olfactory cues to like a puff order and see whether that works similarly and whether you get similar activity?
It's a great question. I think maybe we can talk about that later. The trick, I would imagine, we thought about it, but the reason why we didn't do it is because we are not very good at controlling odor delivery to the animal and making sure that trial by trial that we can change. Exactly. But maybe there's a way to do. I know Dima Rimberg has this particular type of nose poke.
If you're complex, if you know already what you're looking for, I think the bar for being absolutely pure and clean with your odor delivery is a little bit relaxed. I would say if you can find similar activity patterns now with a modality that's not even used in the cueing on the task.
Oh, I see.
The same task, everything is identical, and now just add another cueing and look for
How invariant are... Yeah, I think that's a great idea.
So I apologize, I might have missed this at the beginning, but in some ways, peripherally connected to Florian's question, is there any crosstalk either at the level of thalamus, meaning you said MD is the main input. Are there primary sensory inputs that give a hint, this is the kind of stuff that's coming in, and maybe tweak the central model that you have with small refinements that might even do edge sharpening more? And
Oh,
can
I see.
imagine those kinds of things at multiple levels.
Yeah. So that's the difficulty with... I think it's a great question. I think...
And I should put a fine point
Yeah. So, yeah, absolutely. So, we can confidently rule out a crosstalk through the thalamus because one thing I should have mentioned early on in the talk is that um, the thalamus and the cortex are different in their basic architectural features in that there is no local excitatory, there's no E to E connections in the thalamus as far as we know. So those neurons literally are independent units that just live next to each other. And that might, in the model that we have, that might give a really nice substrate for this contrastive Hebbian learning that the thalamus may be doing. So the neurons can suppress each other through the TRN, and therefore if one unit learns something, it can suppress all the neighbors. That's potentially, you're limited by how many contextual representations you can make by the number of neurons that you have in the thalamus. I mean, that's one potential idea. But so the information from sensory modalities making it to the MD through the thalamus itself is unlikely. But the fact that MD neurons could be sampling from multiple different cortical areas that also process the cue, that's very possible. It's a hard question to be, so it's a very hard question to be able to completely nail. Yeah.
It would be a second or third order term.
It's hard to know, actually. It could be very central, but we haven't figured out exactly the type of experiment to do to rule it in or rule it out.
Is there any layer specificity to the responses in PFC?
Specificity.
Like if you record layer 2, 3, or layer 5.
Oh, the layer specificity. Oh, yeah. So in this task, these neurons, the meaning neurons, the ones that project, at least a subset of those neurons that project to the striatum are infragranular. So they're layer 5, layer 6 neurons, or layer 5 mostly. But the cue-specific responses are distributed all over. So I imagine that they're going to be a mixture between, cortical-cortical, between cortico-thalamic. They have to be cortico-thalamic because they got to make it to the thalamus. And in this task, the striatum. But I imagine that this is just a more general thing. I mean, that's the hope that it would generalize to other tasks. That's what we're testing right now. But I don't know. Oh, sorry.
I have a question about the difficulty signal in MD. So is this signal, so the prefrontal doesn't have difficulty information before the MD, or the MD still get the information from the PFC?
So the PFC does have, so it seems that the PFC has the opposite of that. So we have everything in the cortex as well. Even the contextual signal is in the cortex. But it seems to be much more obvious in the we can decode it with fewer neurons from the thalamus than the cortex. And the predominant sort of ambiguity signal is decoded from the thalamus. In the cortex, it seems to be the opposite signal. It seems to be how confident that the spike rates go up when the stimulus is easy.
So the MD is extracting that information.
That's so through the same sort of circuit diagram. And I mean, this is just a cartoon, right? It's not really a model. But I think it summarizes quite a bit of sort of data, right? It's simply that the MD learns these weights, and therefore the summation has got to be nonlinear, meaning that 2 and 0 is less than 1 and 1. So if you have a nonlinear term in your interactions between the inputs, you would get responses that reflect confidence. Now, exactly how that's biophysically implemented, I don't know, but that would be, that's sort of what we're trying to understand right now.
And then the signal aids the difficult task performance, but it only helps during the difficult trial. So is this signal expensive, or why only?
Well, so again, I mean, this is I apologize. This might have been too fast, but if you get something, so this is basically the cartoon. If you basically have a very, very clear signal here and nothing here, right? Sorry, nothing here, right? You don't need anything to, so what you're trying to avoid is that these less likely hypotheses, right, survive into working memory, right? Because the animal presumably is forming some kind of idea of what the stimulus is based on the cues that are provided. So the engagement here is to kill off the less likely input. When you don't have it provided in the first place, it seems that this would be less likely to engage.
Maybe I ask last question. So, regarding the mechanism of sustaining the sequence of activity in the prefrontal cortex, you are kind of emphasizing local process within DHC, but the cross-correlation analysis or coupling analysis you showed is second time scale, not millisecond time scale. At least one example that you showed.
You're talking about
Coupling.
this stuff?
I think much earlier I thought the
Cross correlation between neurons.
Oh, the cross correlation is 3 milliseconds.
3 milliseconds.
Yeah.
Okay. Oh, that was millisecond.
Yeah. If I labeled it as oh wait, sorry, no, it's here. This one.
Okay. I was wrong.
No, it's okay. Yeah.
So
But-
then
yeah.
you need to have these two populations relatively segregated, right? So that
You mean spatially or functionally?
or in terms of connectivity.
Oh yeah.
Yeah. So that also seems to be the case.
And these things presumably happen through learning. That the segregation of connectivity, this versus that,